Today, we're diving into something that sounds like it's straight out of science fiction, but it's very, very real. We're talking about genetic engineering, how scientists can literally rewrite the code of life itself. So, let's start with a really cool idea. A glowing idea, actually. This story sounds like it's straight out of a movie, but I promise you it's happening in labs as we speak. All right, so the big question is, can you actually give a human gene to something like bacteria? Well, to get there, let's tackle a slightly different question first. What do you think happens when you give bacteria a gene from, say, a glowing jellyfish? And get this, it worked. Scientists literally took the gene that makes a jellyfish glow and popped it into bacteria. And guess what? The bacteria started glowing. I mean, how cool is that? This was huge because it proved you could move a trait from one organism to a totally different one. So, back to our first question. If you can do it with a jellyfish gene, you can absolutely do it with a human one. And this isn't just a party trick. This exact idea is used for something life saving, making insulin for people with diabetes. They give bacteria the human gene for insulin, and boom, you've got these tiny little factories pumping out medicine. So, this whole process, moving genes around, changing an organism's traits on purpose, that's what we call genetic engineering. You're basically getting under the hood and editing an organism's DNA to give it new abilities. Okay, but how do they actually do it? How do you cut and paste DNA? It sounds impossible, right? Well, let's use that insulin example and break down the recipe. It's kind of like a high tech cooking show for DNA. First things first, you need a way to get the human gene into the bacteria. You need a delivery vehicle, or a vector as scientists call it. And for that, they often use something called plasmid. Just think of it like a little DNA shuttle bus that bacteria are totally cool with letting inside. All right, so you've got your shuttle bus, but you need to make a space for the passenger, the new gene. That's where restriction enzymes come in. These things are incredible. They're basically molecular scissors that snip the plasmid DNA right at a specific spot to open it up. And of course, if you have molecular scissors, you're going to need some molecular glue, right? So once you've inserted the human insulin gene into that open spot on the plasmid, another enzyme called DNA ligase comes in and seals it all up. It glues the ends together, making it one solid piece. So there you have it. That's the basic recipe. Step 1, get the gene you want. 2, cut your plasmid open with your molecular scissors. 3, pop the new gene in. And 4, glue it shut. A really clever part of this is that they use the same enzyme to cut both pieces of DNA, so the ends are sticky and match up perfectly. It's really elegant. And what you end up with is recombinant DNA. It's a brand new hybrid piece of DNA that's part bacteria, part human. A combination, or recombination, of two different sources. Pretty wild. Okay, last step. You've made your custom DNA, now you just need to get the bacteria to pick it up. This step is called transformation. And once they take in that new plasmid, the bacteria do what they do best, multiply. And they multiply fast. So pretty soon you've got millions, even billions of tiny little insulin factories all working for you. But, as cool as molecular scissors and glue are, that's kind of old school tech now. The genetic engineer's toolbox has gotten a major upgrade over the years. It's packed with all sorts of powerful new tools. The big one you've probably heard of is CRISPR. It's a total game changer. Think of it like this. A restriction enzyme is like the find command in a document. It finds every instance of a word and cuts it. But CRISPR, that's like find and replace. You can tell it exactly which specific word in which specific sentence you want to change. It's incredibly precise. And the toolbox just keeps getting cooler. Scientists can even hijack viruses, since viruses are naturally amazing at injecting their own DNA into cells, and use them as delivery drones. Or there is microinjection, where you literally use a microscopic needle to inject a gene. And my personal favorite, which sounds like something out of a sci-fi movie, is the gene gun. Yep, it literally shoots tiny gold particles coated in DNA right into a cell. So this is the part that really matters. This isn't just about cool experiments in a lab. All this tech, these tools, they're out in the real world, and they are changing everything. I mean, the applications are just huge. You've got medicine, where we're making not just insulin, but things like clotting factors for hemophiliacs. In agriculture, we're making crops that can fight off bugs or survive with less water. For research, we can modify animals, like mice, to help us understand human diseases better. And we're even looking at engineering plants that can literally suck pollutants out of the ground. It's incredible.
So, with all this incredible power, you can see how the conversation starts to shift. It's not really a question of can we do this anymore? Now, the really hard question is should we?
And that's where things get really complex, right? We have to start talking about the ethics of it all. There are no easy answers here. We're talking about big things like animal welfare, what happens if we accidentally release a modified organism into the wild, and, who gets access to this technology? Is it for everyone or just the wealthy? These are serious conversations we need to have.
So, I'll leave you with this final thought to chew on. For the first time in history, we have the tools to actually steer the course of evolution. We can rewrite the code of life. And as that power grows, well, so does our responsibility. We have to be thoughtful, we have to be wise, because what comes next, that's a future we have to decide on together.